sizes, but also the plants themselves. I mean, we have maize teosinte plants that can be only a foot tall and up to 25-foot tall plants, variation in the number of tillers and purple to green, all sorts of phenotypic variation. And as I moved forward in my career, I did a PhD in genetics and started thinking about evolutionary biology. And I remained fascinated with maize because of its tremendous genetic diversity as well. And this is one of my favorite displays of it. So each line here is a maize individual with 2 copies of each of its 10 chromosomes. So there are 20 chromosomes. And this is fluorescent in situ hybridization. So we take oligos that match some repeat and hybridize those to the genome, and then you can look at them fluorescently. And this is work by Jim Birchler's lab. But the neat thing here, each color is a different kind of repeat. And you can just visually see the tremendous diversity across chromosomes, across individuals within a chromosome. See, yeah, so here, so across individuals within a chromosome, within individuals that are heterozygous for these, there's just a tremendous sort of variation in these repeats that you can just visually stare at. To maybe put those into more concrete numbers, if we think about just the number of SNPs or mutations, if we compare any 2 people on this call, we differ in something like 3 million mutations. So any 2 random people differ by something like 3 million mutations. If we compare a human to a chimpanzee, those differ, any one of us differs by about, on average, 30 million mutations between humans and chimpanzees. And if we compare B73 to Mo17, or any 2 sort of modern inbred varieties, maybe not varieties from the same heterotic group, those differ at something like 60 million mutations. So an order of magnitude more variation than you see in something boring like humans. And this diversity has really gotten me excited and kept me excited about maize as a system. But I'm also an evolutionary biologist, and one of the great things about working on a domesticate is domestication, I think, is a wonderful analogy for evolution. And of course, I'm not the first person to have thought of this. Domestication and variation in domesticated plants and animals really motivated Darwin in his Origin of the Species, arguing that a careful study of domesticated animals and plants offers the best chance of making out this obscure problem of evolution. And he argues that really there's tremendous similarities between selection under domestication and under cultivation and natural selection. So he calls this unconscious selection, which is when people naturally preserve the most valued and destroy the less valued individuals without any thought of altering the breed. So over thousands of years of selection by indigenous farmers or even modern day farmers, there's a lot of the selection is I'm picking the best plant and keeping the best plant, not because I'm specifically trying to directly or modify a particular phenotype in some direction. Obviously, in modern breeding, there is some of that, but throughout most of the thousands of years of selection during domestication and cultivation, most of the selection is you're picking the best plant, not trying to pick specific phenotypes. And one of my favorite examples of that in modern corn breeding is leaf angle. I'm certainly, maybe a few breeders have selected specifically on leaf angle, but I'm not aware of much active selection directly on leaf angle. People have selected for yield per acre, for example. But as a result of selection for increasing yield per acre, one of the phenotypes that has changed most dramatically in the last 40 or even 80 years of maize breeding has been leaf angle. Again, not because someone has directly said, I'm going to select on plants with higher leaf angle, but because selection on yield per acre has given us plants that have higher leaf angle so that you can pack in plants more densely and shading isn't a problem, et cetera. And in fact, a really nice meta-analysis of the rate of phenotypic change under unconscious selection in domesticated plants and natural selection in wild plants shows that actually the rate of phenotypic change in domesticated plants, if anything, is slightly smaller than under natural populations. So the idea that selection during domestication and cultivation is somehow aberrant or unusual doesn't really hold up. And in fact, it's this unconscious selection, which I would say has been responsible for most of maize adaptation, right? So maize has adapted to, if you sort of divide up the world into grid cells, maize is adapted to a broader array of grid cells, a broader geography than any other cultivar. than any other cultivated plant, from southern Helsinki to the equator, and from sea level to, we're working on some maize in Tibet that grows almost to 5,000 meters above sea level. So it's adapted to an enormous range of environments, predominantly through this process of unconscious selection. And so maize is a really wonderful model for thinking about how adaptation and evolution occurs. And indeed, a bunch of topics in evolutionary biology we've learned about directly from maize, whether that's inbreeding depression. So this is work, Charles Darwin was the first to do work on inbreeding depression with a number of different plants. And this is his data in maize. And each column here is a pair, an outcross and self sibs from a family, showing that on general, the outcross plants are taller and happier than the inbred plants. So some of the first work on inbreeding depression 150 years ago was coming from corn. Obviously, more modern things like our understanding of transposable elements and Barbara McClintock's work on meiotic drive, so selfish elements that sort of cheat at meiosis, has come directly from maize, from the work of Rhodes and others. Some of the work on actually how populations evolve and how populations stay separate, this idea of reinforcement, has come from maize. And there's beautiful experimental work showing that you can keep these maize populations separate by selecting for decreased hybridization between populations, inadvertently selects for changes in flowering time. And so after several generations of selection, these authors show we get a shift in earlier flowering and later flowering in two different populations to minimize hybridization. And then more recent work, for example, our understanding of genetic architecture of traits, a lot of that has come from the wonderful resources we have for maize and the sort of high resolution mapping that we can do. One of the best things about thinking about domesticates as a model for evolution is the fact that for most domesticates, we have this really clear ancestor that we can compare to. In a lot of evolutionary scenarios, I go out and look at some finches on an island in Galapagos, and I don't know what they looked like 10,000 years ago. But for most domesticates, we can compare directly to a wild ancestor, and we can, by that comparison, we can learn about what phenotypes have changed and understand the genetic basis of that and how evolution has occurred. And this, my favorite example of this is all of the different domesticates that have come out of the same wild brassica. And by comparing the phenotypes, you can understand what parts of the plants have changed and then think about what genes have changed. Unfortunately, in maize, one of the, I think, two sort of embarrassing secrets of maize domestication is that for the last 120, 150 years, we haven't known what the wild ancestor of maize is. And that's largely because maize is pretty weird looking. Maize is a grass, but it doesn't look like what most people sort of think about as what your idea of what a grass looks like, something like a prairie grass or a lawn. And this led to lots of confusion about even what other species could potentially be related to maize. And even by the middle of the 20th century, botanists like Edgar Anderson weren't actually sure what continent maize came from, because it was so different from all the possible wild relatives that he couldn't even narrow it down to a continent. And in fact, we know now that even comparing maize to what we know is its closest wild relative, teosinte, they're really dramatically different. So dramatically different in overall sort of architecture of the plant. Maize has a single central stalk with one ear. Teosinte has multiple tillers. Some of these stalks have branches on them. The lateral branch in maize ends in a female inflorescence, and in teosinte ends in a male inflorescence. And then the most notable, of course, are these massive differences in the ear, whereas maize often will have one ear with hundreds of kernels that are attached to the cob and remain so at maturity and are uncovered. In teosinte, you have these tiny ears of 8 to 10 kernels. There are hundreds of ears on a plant. And at maturity, the seeds mature and dehiss or fall off, so they just scatter to the ground. Moreover, each kernel is actually tightly encased in a fruit case that is so tight, so hard that you could sort of break a tooth on it. So these really dramatic morphological differences between maize and even what we know are its closest wild relative. And this is what led to lots and lots of confusion about the origins of maize and how you could possibly get this thing on the right from anything else. Throughout most of the 20th century, actually the most widely accepted model, based on a lot of work in the archaeological record, was a sort of complex model. The authors argued that we could find in the archaeological record, we have this sort of ancient maize, and over time in archaeology, we see that the maize cobs get bigger, and the morphology changes some, which Mangelsdorf and other authors argued was a result of hybridization that increased vigor in maize. And they argued that this model made sense because they could make these sort of cross-generic hybrids between maize and other grasses that had some of this odd morphologies that matched things in the archaeological record. And so the model that Mangelsdorf and others came up with seems a little bit silly in retrospect, but they argued that there was some perennial grass, which nobody has ever identified, so an unknown perennial grass, that hybridized with some extinct maize, which nobody has ever found and there's no evidence existed. So an unknown perennial crossed with an extinct thing, hypothetical extinct maize, and that hybrid gave rise both to modern maize and teosinte. And so we have these sort of two mythical progenitors of both modern maize and teosinte. What we know now is, or much closer to what we know now is the model, is a much simpler model put forward by George Beadle. George actually did his PhD in maize genetics and then went on to waste most of his career on Ross-Ibara and something about genetics and a Nobel Prize. But after he retired, he actually came back to work again on this question of maize genetics and origins. And Beadle argued for a fairly simple model that maize was domesticated from something like teosinte. And he did this by making crosses and looking at chromosomes and showing that maize chromosomes don't associate with chromosomes from some of these other perennial species. He did this by showing that you could make popcorn from teosinte. And he did this by grinding up teosinte and making these really gross tortillas that are sort of like a tortilla with sand in it that he would take around on his field excursions or take to seminars and sort of force feed to people to prove to them, look, I can make tortillas from teosinte. And so his model was a relatively simple one, that some ancestral teosinte gave rise to both modern teosinte and modern maize. Now, when I say teosinte, teosinte is a generic term for wild species in the genus Zea. And there are a number of these species that occur in Mexico and Central America, but they don't occur in South America or farther north than Mexico. But they occur in a wide range of environments, from waterlogged conditions in Central America to arid mountainous regions in central Mexico. And they differ in a whole range of phenotypes, from neat Erynchyma cells and Zea luxurians to perennial taxa like Zea perennis and Diploperennis that have these perennial rhizomes. There are tetraploid species, et cetera. So there's a tremendous diversity of these teosintes. But most authors largely recognize that there are two teosintes, two, what we now call subspecies of Zea maize, subspecies parviglumis and mexicana, that were the most likely contenders for the wild, or the ancestor of modern maize. And both of these had proponents, and in fact, a lot of the early domestication literature involved crosses to different accessions of both of these teosintes. I'll use these red and blue icons throughout the talk to show these two teosintes. Zea maize subspecies parviglumis grows in the lower slopes of the Sierra Madre in Mexico and Southwest Mexico and hotter conditions along the coast, and occurs in these very large populations. So all of the grass here on the bottom left that you see, each of those grass plants is a teosinte. Mexicana grows in the highlands, so I'll refer to it as highland teosinte, at elevations above about 2,000 meters in the highlands of the Central Plateau, and occurs in generally smaller populations as sort of a weed in fields and along roadsides. On the bottom right is my favorite picture of Mexicana. This is a teosinte plant growing in Norman Borlaug's wheat field, sort of showing all the wheat plants who's boss. These two taxa, Mexicana and parviglumis, diverged about 30,000 generations ago. And so maybe one way to think about that is in terms of generations, that's about the same as the separation between humans and Neanderthals. So they're fairly strong, they're fairly deeply diverged, and they both have adaptations to their different environments, a number of physiological, flowering time, and morphological adaptations. So on the top right, these Mexicana plants tend to be darker colored and have hairs on their leaf sheaths and sometimes on the stem, which are adaptations to colder conditions. and you see these glabrous, greener plants in parviglumis. And the first sort of effort to disentangle which of these was the most likely wild relative was done by John Dobley in the early 2000s. And what John did is John took a large number of microsatellite loci of early genetic markers, and he just built an evolutionary tree of hundreds of maize and several of parviglumis and mexicana. And what John showed was that all maize formed a single clade together, so they all grouped together, and they all grouped together sort of nesting within this parviglumis. And so John's group made the claim that thus we have a single simple domestication of some ancestral parviglumis that gives rise to the modern parviglumis we see today, and as well as modern maize. And this has really been the dogma for the last about 20 years, this simple single domestication of maize from parviglumis. But when I started working on maize genetics about 15 years ago, it became clear from a read of this paper that there was a conflict, or essentially a conflict between the genetic data and the geography and ecology, such that if you take these samples that I've circled here of maize, these are the maize samples that genetically look most similar to teosinte, and you ask where are they from, so they look most similar to parviglumis, but they're from the highlands of central Mexico. So they're from the wrong part of the geography, from the wrong part of the range, and they're pretty distant from the oldest archaeological evidence of maize domestication. So the maize plants that look most genetically similar to parviglumis are far away from parviglumis, within the range of this highland teosinte, and far away from the archaeology. And that conflict was something that we were quite interested in. And over the last 10 years, with a group of researchers at North Carolina and Langebio in Mexico and USDA and Iowa State, we've done quite a bit of work to understand how maize is adapted to high elevation conditions worldwide. And in the process of that work, we found quite a bit of evidence of contributions for the second teosinte, this highland mexicana. So for example, we first started asking, how does maize adapt to these highlands of central Mexico? And if you go even today into traditional maize fields in the highlands of central Mexico, you see maize here on the left, and on the right is a small population of mexicana. So these things grow meters apart. You can find hybrids in farmers' fields. Farmers complain about it because it's hard to weed out the teosinte. And if you look at the adaptations of highland maize versus lowland maize here on the left, highland maize differs from lowland maize in a lot of parallel ways of mexicana differing from parviglumis, the stem pigmentation, the pubescence, flowering time, and a number of other traits. And we were able to show that a bunch of those phenotypes arose thanks to adaptive introgression from teosinte. So maize was actually stealing genes from highland teosinte via gene flow, and those selection by indigenous farmers over hundreds or thousands of years favored those alleles and allowed maize to adapt to the highlands of central Mexico using these alleles from mexicana. We next, a few years later, looked at how maize made it into the US and using genetic analysis of ancient DNA of cobs in caves from Southwest US, we could sort of partition how much of the genome of these ancient and modern varieties come from low elevation, high elevation, or teosinte. And what we found is that the oldest cobs, 4,000-year-old cobs in these caves in the Southwest US, so the very first archaeological evidence of maize in the US, came from the highlands of central Mexico. So maize moved up from sort of the central highlands of Mexico into the Southwest, bringing with it alleles from this highland teosinte mexicana. And then most recently, and what really sort of set us down this path, is we were interested in thinking about how maize adapted to the Andes. And we thought this was a neat comparison because there is no teosinte in South America. So we thought there's no way that teosinte could contribute to highland adaptation in the Andes. And yet we see maize growing at elevations of 3,000 meters and higher in the Andes. And using population genetic approaches, we could sort of scan along the genome and identify regions like I've shown here in this box, that show evidence of selection both in Mexico and in South America. And we were interested in sort of whether there's evidence for lots of these regions and how convergent evolution was. And what we found, there was lots of selection that's sort of unique to the Andes. About 10% of the genes that show evidence of selection in the Andes also show evidence in Mexico. And then we can use some modeling approaches to ask, okay, if they're selecting on the same gene, is that because they have independent mutations, or there was some ancestral variation they were selecting on, or was there movement of the gene from one to the other? And invariably, we found that when they share selection on the same gene, it was almost always the case that there was evidence that that gene had been selected in Mexico and moved from Mexico to the Andes. So beneficial highland alleles from Mexico moving all the way down to the Andes, carrying with them this highland teosinte ancestry. And this really surprised us. We now see highland teosinte ancestry everywhere when we look at the highlands, and we were wondering, does this argue that there's a broader contribution of this second teosinte, this highland mexicana, or is it just for adaptive loci in highland regions? And we had some reason to doubt that this would be sort of widely, that there would be a lot of gene flow from the second mexicana, because there are a whole bunch of genetic barriers in maize that prevent gene flow from teosinte. There's a set of loci referred to as the GA loci, GA1, TCB1, GA2, and these are pairs of proteins that act in pollen-pistil interaction. So pollen lands on the pistil, has to germinate, and the pollen tube has to go down to fertilize the ovule. And if you have the wrong combination of these genes, then you're not able to pollinate individuals with a different combination of those genes. And so there are active barriers of these barriers in many teosinte populations that actually prevent maize pollen from pollinating and fertilizing teosinte. So this is a barrier to hybridization between these taxa. And more recently, this past year, in work with Rob Martinson's lab, we published work on a complicated pollen killer locus. So this is a set of loci that I'll talk a bit more about at the end, that in hybrids between maize and teosinte, these pollen killer loci end up killing 3/4 of the pollen and only allowing pollen with the teosinte mexicana alleles to survive. Again, seeming like be not a great solution for, or a great way to allow hybridization if all of your hybrids suddenly have low pollen fitness and don't produce a bunch of pollen. So we thought that these barriers would likely prevent widespread gene flow between maize and these other teosintes. But what we didn't have was good data to sort of assess this. And so starting a couple of years ago, in collaboration with Jianbing Yan at Huazhong Agriculture University and a project led by my postdoc Ming Yang, who's now back in Huazhong Agriculture University as a professor, we did what my lab tends to do. We sequenced everything we could get our hands on. And so we got our hands on as much teosinte seed from as many different taxa as we could and sequenced everything. So we ended up with hundreds of wild teosinte, hundreds of modern inbreds, and several hundred traditional maize varieties collected throughout Mexico to get a sense of how much gene flow is actually occurring and how much of the genome is impacted by this gene flow. And can we learn more about the contribution of the second teosinte? And the way we did this is a very simple sort of tree test. So if we imagine that this tree in the middle this is essentially the tree that John Dobley's lab argued for. And if this is the correct tree, we can use this statistic F4, which basically looks at the covariance of alleles along a tree. And if this tree is correct, F4 gives us a value of 0. If there's been gene flow between the two teosintes, we'll get a negative value of this F4 because it changes how allele frequencies will covary. And if there's been gene flow from highland teosinte into maize, as the tree on the right shows, we'll get positive values of this F4 statistic. And when we start taking all of these sequence genomes that we look at and evaluating this F4 statistic, we see that in the 3 cases I just told you about, the highland, Mexico, Andes in the Southwest, we see strong evidence of gene flow from mexicana, so there's positive values of that F4 statistic. And there you go. But when we start looking at our other taxa as well, across all traditional varieties of Mexico, in Mexico, we see positive values of F4. Across traditional varieties from China, we see positive values. And in every single one of the modern inbreds we looked at, we also see positive values of F4, consistent with widespread gene flow from mexicana into all maize. And in fact, we can show that this is even more abundant than those data suggest by looking at genotyping data from more than 5,000 traditional maize varieties across Americas. We see that all of them have non-zero levels of admixture from the second teosinte. And if we just do a principal component analysis on genetic diversity in maize and ask what are the factors that most explain maize genetic diversity, we find that the first principal component perfectly correlates with how much mexicana admixture. So that's saying that gene flow from this highland teosinte in regions as far away as Brazil and the Andes still is the major component, the single major component that explaining variation in maize diversity across the Americas. Given that we see ubiquitous gene flow, we then turned to looking at, could we say when this happened? And to do so, we can take each one of these genomes and we can go along the genome with some statistical models and say, this piece of the genome comes from Mexicana, this piece of the genome comes from lowland teosinte. So we can go along a genome of maize or teosinte or any plant and say, highland teosinte region, lowland teosinte region. And this is what it looks like for the inbred B73, where the y-axis is the probability of this region coming from highland teosinte. And you can see that there are a number of such regions that have this high probability, but they're all pretty small. So they're all pretty small admixture blocks, about 10,000 base pairs on average. And we can use the size of these blocks and the abundance of these blocks to actually estimate the date of when this gene flow occurred. And our estimated date is about 5,700 years ago, which corresponds really nicely with the earliest evidence of archaeological maize in the highlands. So the earliest archaeology says that maize wasn't even in the highlands of central Mexico until 6,000 years ago. And shortly thereafter, the population genetic data suggests that there was lots of hybridization. We can confirm this by looking at ancient DNA. So if we plot on our F4 statistic for ancient DNA from South America, that's almost 1,000 years old, or 5,000-year-old cob from central Mexico, in all of these, we see positive values of F4. But we were most excited about this really neat cob that was just published a year ago from Peru. And this cob is an almost 6,000-year-old cob. So this maize was already in South America. And as you can see from the picture, it's clearly a domesticated maize. It was already in South America before there was any possible hybridization between maize and highland teosinte, because maize had just made it to the highlands of central Mexico. And so this cob, a priori, should have no evidence of gene flow from the second teosinte. And in fact, it's the only sample that we've looked at across more than almost 1,500 maize and teosinte samples that has zero evidence of gene flow. So that simple tree of direct domestication from parviglumis fits nicely. So this is consistent with this gene flow happened in the highlands of Mexico after domestication. We can sort of put this all together, and I won't walk through this in detail, but we can put this together into a complex model of how maize domesticated. We get the split of this Peruvian lineage, hybridization in the highlands of central Mexico. And then as maize moves out of the highlands of central Mexico into the lowlands, it again mixes or hybridizes with the lowland teosinte and then spreads across Americas, such that about 20 to 25% of any given maize genome comes from the second teosinte. And this is across all maize inbreds we've looked at from across the world, from across different breeding programs, including elite material. About 20% of the genome from different modern maize comes from the second teosinte mexicana. And this is in comparison to, for example, if we think about Neanderthal admixture in humans, it's only 2 to 3% of our genome comes from Neanderthals. Okay, so there's widespread gene flow between maize and mexicana. This mexicana, the second teosinte, has contributed a significant proportion of the genome. But I told you several slides ago that we had these genetic barriers that should prevent such gene flow. And so work that hopefully will be online next week by my grad student, Ellie Cryan, has looked at, for example, these genetic barriers, these pollen pistil barriers between maize and teosinte. And what she finds is that in many teosinte, we have normal functional copies of these alleles. So we have functional copies of these barriers that prevent hybridization from individuals that don't have functional copies. But in many maize plants, including, for example, the inbred B73, in many maize plants, at one of these loci, GA1, we find this really strange pseudogene array. So an array of non-functional copies of this gene, as many as 50 copies, non-functional copies. And that haplotype is at very high frequency in maize. About 60% of modern maize has this array of non-functional copies of it. And what Ellie was able to show is that this non-functional copy actually generates small RNAs, and those small RNAs target all of these loci and silence them, actually turning off all of these loci, so that the non-functional haplotype in maize actually overcomes the barrier by silencing all of these genes, just methylating them and turning them off. And she's been able to show this by looking at small RNA and also by doing a nice set of crosses where she can take an active barrier. So here she's crossing colored pollen on and yellow pollen in competition onto a plant that has an active barrier, and we get no colored kernels. But then she takes this active barrier and the same exact sequence, she just puts it in a hybrid with a non-functional array. And if our hypothesis is right, that array should silence the active barrier and turn it off. And sure enough, in that hybrid, our silenced locus now accepts colored pollen, proving that we've turned off the barrier, even though the sequence of the locus hasn't changed. So we have this barrier, the barrier really exists, but Maize has evolved the locus to sort of overcome the barrier and allow for gene flow. I also introduced this pollen killer locus. And this is work by Ben Berube that was published just last year that we collaborated with Rob Martinsen's lab on. And the way this works is there's a locus called TDR1, and TDR1 just makes a pollen lipase. It's a protein that is necessary for normal pollen function. But there's a second locus, TPD1, which makes a hairpin RNA. That hairpin RNA targets TDR1 and turns it off. If you don't make any of the pollen lipase isn't translated, your pollen dies. In teosinte, there's a locus TPD2, which downregulates TPD1, so shuts off this pollen killer function, and the pollen is able to make normal lipase, and teosinte plants crossed with teosinte are just fine because they have this locus which turns off the pollen killer. Maize doesn't have TPD2, and so then you get, in hybrids between maize and teosinte, you get this pollen killer effect that you lose 75% of your pollen. But what Maize has done instead is said, instead of trying to worry about this second TPD locus, Maize has actually evolved immunity to this pollen killer. And it's done so by just a simple deletion in the lipase gene, such that the small RNA which targets the lipase can't bind anymore because of this deletion. It's an in-frame deletion, so the protein is totally functional, but is unable to be silenced or silenced by this pollen killer locus. In teosinte, this deletion in orange is at super low frequency, but as you move to traditional varieties, it's up to 81% frequency, and it's essentially at 100% frequency when you get to modern inbreds like the stiff stock heterotic group. So there's clearly been selection in maize for this deletion to prevent the pollen killer effect and allow hybridization with teosinte. So in both of these systems, we have these systems that prevent hybridization, and yet maize has evolved to overcome these systems and allow hybridization. So our model is now that maize was originally domesticated and dispersed from the lowlands, and this happened sometime before 6,000 years ago, and this is consistent with, say, John's genetics and George Beadle's cytogenetics. And sometime after 6,000 years ago, as maize hybridized with mexicana in the highlands of central Mexico, it then dispersed back across the Americas, and this new maize mexicana hybrid replaced everything that was previously there. And this is consistent with our new genetic data, as well as that early archaeological data I told you about, where the cobs show evidence of admixture and increases in vigor. And this now led to the big question that we just hired a postdoc to keep working on, is that why did this happen? So why was this admixture beneficial? And we think that maybe this admixture was actually important in making maize a staple crop. So maize was domesticated about 9,000 years ago. The admixture happened about 6,000 years ago. And if we look at carbon isotopes from human skeletons across the Americas, you can actually tell how much maize they were eating by the ratio by looking at C14 isotope data. And if we look a few hundred years, less than 1,000 years after this admixture event, as maize is spreading across the Americas, maize suddenly becomes a staple crop across most of the Americas. And so while that's sort of circumstantial evidence, it suggested to us that maybe there's something here that the admixture with mexicana was beneficial for maize. And so I don't have, and then clearly after that, you have the rise of agricultural villages and the first regional civilizations, things like the Maya and the Aztecs that you're familiar with. And while we don't have definitive answers to this, we have some work underway that provides at least some tantalizing clues as to why this might be beneficial. So we know if you make modern maize mexicana hybrids, they're pretty darn vigorous plants. They're huge compared to maize plants. And we thought that perhaps, and I think the best model for why this admixture may have been beneficial is that deleterious alleles, or essentially bad mutations or genetic load that accumulated during domestication, were complemented by alleles from the second teosinte. So essentially, we have something akin to heterosis by crossing what had become a somewhat inbred domesticated maize with this really diverse wild relative. And to test that, we looked at regions of the genome where we have DNA from the second ancestor and regions where we don't have any admixture. And we can see that regions with admixture have many fewer deleterious alleles than other regions, consistent with gene flow from teosinte was basically complementing load that had accumulated in modern maize. But we can also do work to identify individual genes that may have been beneficial. So for example, our work in the highlands of Mexico, by looking at QTL mapping and some population genetics, we can find a really small QTL for this sheath, this leaf hair, stem pubescence phenotype. And assemblies of highland and lowland maize genomes find an inversion right inside this region. And right in that region are a small number of genes, including a classical maize mutant macrohairless 1. So we are able to identify this macrohairless 1 gene shows evidence of selection in the highlands for the formation of more stem pubescence. By just looking at which regions of the genome show most mexicana alleles, which is graph here on the top, we just sort of look along the genome and say, gee whiz, here we have almost every maize line has a mexicana allele at this part of the genome. Let's look and see what those genes are. If we look in, for example, B73, this track of Mexican ancestry shows sort of a window right around a single gene. So we don't even have to ask which gene in this QTL. There's only one gene in this admixture track. That gene clearly was inherited from Mexicana. That gene, PR37A, is known to be in the circadian rhythm pathway. And work from collaborators knocking out this gene, we can show that knockouts of this gene have a dramatic effect 5 to 10 days on flowering time in long day conditions, but knockouts of this gene have zero effect on flowering time in short day conditions, consistent with its role in flowering time. And we think an adaptation as maize moved out of the tropics to higher latitudes and had to adapt to short day conditions. In addition to these sort of pop-gen approaches, we have work underway trying to map phenotypic basis for some of these loci. So here we're doing sort of standard GWAS approaches, but instead of using the genotype at each locus, we use ancestry. This is sometimes referred to as admixture mapping or ancestry mapping. So each individual at each location along the genome, you get a 1 if you have the Mexicana allele, and you get a 0 if you have the normal maize allele. And then we can do association mapping with that ancestry genotype. And when we do that, we find loci like DGAT1, which is known to be important in triacylglyceride synthesis for oil content in kernels, and we think maybe freeze tolerance at high elevation. And we find an intriguing locus that's still uncharacterized that is really strongly associated with cob diameter, and maybe one of these loci that could have contributed to increased maize yield as a result of admixture from Mexicana. And finally, we can take sort of a step back and take a broader quantitative genetic approach and just say, in a mapping population of inbreds, if we characterize these inbreds as Mexicana or maize, how much of the additive genetic variation for agronomic traits we care about is due to variation coming from Mexicana? And we see across several broad categories of agronomic traits, plant morphology, biochemical traits, and disease resistance, that although it's not always super high, there are some that are pretty striking. So for example, a quarter of the additive genetic variance for kernel row number comes from variation in modern maize inbred lines for whether or not they have the Mexicana allele. We see contribution to some of these biochemical traits, and for some disease traits, for example, maize rough dwarf virus, half of the additive genetic variation for resistance comes from segregation of Mexicana alleles in modern inbred lines. And we think, what I'll end with here is we think that this actually suggests that sort of an interesting way of thinking about incorporating novel diversity into maize breeding programs. There's lots of talk about crop wild relatives and using alleles directly from crop wild relatives in modern varieties, but as lots of us on this call know, that's very hard. Nobody wants to introgress a teosinte into a modern elite. But what we've shown here is that actually there's been lots of historical integration of teosinte or wild relatives into traditional varieties. And there, one of the big advantages is thousands of years of selection by indigenous farmers has actually done quite a lot of the hard work for us by, for example, taking a historical introgression where there's an adaptive allele linked to deleterious alleles and removing a lot of those deleterious alleles, such that we now have in traditional varieties of maize adaptive alleles from teosinte that have already been selected on. We know they work well in a maize background, and a lot of the sort of genetic load has been removed. So that probably we could save a lot of time and effort, not going directly into crop wild relatives, but surveying the really tremendous variation we have in traditional varieties of maize grown around the world that have already incorporated a lot of that crop wild diversity. Okay, I'll end there. So we have hopefully a few minutes for questions. I've shown lots of admixture from the second teosinte into modern maize. That this gives us a new model of how maize originated. We have sort of two waves of maize migration out of Mexico. And that this has done so in spite of these interesting genetic barriers to hybridization, and may have contributed to maize as a staple crop through a number of both additive genetic variants and individual loci. Lots of folks to thank: UC Davis and abroad, USDA, Wahzhong Agriculture University, and lots of collaborators, as well as a number of sources of funding over the years. Of course, need to thank the folks in my lab who've put up with me and have done a bunch of this work. And with that, I'd be happy to take any questions.
Thank you, Jeff. That was a fantastic presentation. Anyone, if you have any questions, please put them in the chat and we'll try and get to them. There is one question already in the chat that maybe you could answer. It is, can we say that adaptation and evolution occurs concurrently, or does one influence the other?
Yeah, so I think adaptation is really sort of a subset of evolution. Evolution is really just broadly change in allele frequencies and can be done through genetic drift, it can be mutation, and it can be adaptive forces like selection. So I would say that they're happening at the same time, but yes, of course, they influence each other, that novel mutations can influence how selection occurs. Selection is going to affect how much diversity is available for subsequent adaptation and so on.
Thank you. We have another one in the chat. Could you share your insights on the evolution of maize traditional varieties and the relationship between these traditional varieties and the earlier forms of maize following the domestication events and modern maize?
Yeah, so I think maize traditional varieties are actually, I think, super exciting because they allow sort of thousands of years of a natural experiment of taking a plant like maize and putting it in new environments and allowing it to adapt to these new environments. We don't know a lot about sort of the proto-maize directly following domestication because we just don't have genetic data and we don't have archaeological remains. But we are beginning to learn a lot about how maize has adapted to the sort of huge array of environments that it occurs in today, both adaptation to climatic conditions, as well as to biotic conditions, as well as there's lots of interesting adaptation to the sort of human environment, individual indigenous groups selecting for a particular color or particular kernel texture for a particular kind of food. And so it's a really amazing amount of diversity in maize traditional varieties, and we have sort of indigenous farmers to thank for doing thousands of years of that selection experiment for us.
The next question is, there's some confusion regarding the use of the term land races, as it can refer to either types of maize or traditional varieties. Could you please share your thoughts on the appropriate use of the term land race / traditional?
We're actually working on a paper on this. I have sort of strong opinions on this. If you go back historically, the term or the use of races of maize actually comes directly out of some of the eugenics ideas in the early 20th century, where geneticists thought that the races of maize pretty well mirrored races in humans. And as we know in humans, and it's also true in maize, the races are described based on a few very sort of superficial morphological characteristics, the shape of the cob or the color of the cob, and really have almost zero predictive ability about the underlying genetic variation. If you sort of divide up maize by their landrace name, the landrace explains almost none of the genetic diversity. And so I don't think it's a useful term, and I think it's a holdover from sort of this eugenic idea of racial types. And so we have abandoned the term entirely to use this idea of traditional varieties. And that, there certainly are varieties that farmers will identify and care about because of the kernel texture or the cob shape or the flowering time. But usually the aspects that they care about explain such a small proportion of the genetic diversity that it's not meaningful in any biological way or thinking about genetic variation to call those as something like a race. And so I prefer the term traditional varieties or morphotypes or something like that.
Thanks, Jeff. The next question is, how do alleles like GA1M and TCB1M factor into overcoming genetic barriers?
Yeah, so at these genetic incompatibility loci, you tend to have a tightly linked male and female factor. And so you need to have, if the female barrier is present, then you need to have the male factor, this TCB1M or GA1M, in order to overcome the factor. And so because the male factor is largely absent in most maize populations, it was thought that that should prevent hybridization. We've done some work to show that actually that male factor can sort of leak into maize populations and can be selected for in maize populations. But what we've shown here is that actually it may not be as important as we thought, because there's this alternative mechanism of this non-functional allele, which can just silence the female factor completely.
Thank you. The next question is, they would like information about the role of Zea nicaragensis and shaping early maize development. If I said that right.
So Zea nicaragensis is one of the species of teosinte. It's found in mostly Nicaragua, Honduras, and Guatemala. We don't know a lot about it. I don't think that it had a major role in maize origins. It probably did, along with Zea luxurians, contribute to adaptation to some of the sort of swampy conditions in Central America. Nicaraguensis and luxurians can grow in waterlogged conditions thanks to these cool parenchyma cells in their roots. And so that probably has contributed to local varieties that are adapted, but I don't think it played an overall major role.
Thank you. The next question is the original maize, no Mexican gene flow, still grown somewhere on Earth, or are all maize in the world contains Mexicana genes?
That was what surprised us. As far as we can tell, all maize everywhere contains Mexicana genes. Now, that doesn't mean that at every region of the genome, in every maize plant, there's a Mexicana allele. So 80% of most maize plants, most of the genes come from parviglumis. And which genes come from Mexicana differs depending on which maize variety you look at. But there are no maize varieties that we've been able to find anywhere, from traditional varieties from China to modern inbred lines, the XPVPs, that have no Mexicana alleles. So the Mexicana admixed maize seems to have completely replaced any sort of proto-domesticated maize everywhere across the Americas.
Thank you, Jeff. Somebody have any other questions? You can type them in the chat. I actually have one myself. Have you done any comparison between, the frequency of some of these adaptation genes that you've seen in some of the traditional varieties compared to some of maybe more our founder lines that we think of modern maize?
Yeah, we've done a little bit of work on that. And so we've thought about this some for flowering time loci. So there are some of these. So for example, this flowering time gene that we worked on and did CRISPR on is found in about 80% of modern inbred lines have the Mexicana allele. And so it's clearly been, or we argue it's been selected for even in modern germplasm. We've done less with some of these other loci. We have work ongoing right now to look at traditional varieties of maize and look for adaptive loci and see, have those been included in modern germplasm? If not, could they be incorporated into modern germplasm? So I think there's a lot more to do there. I think that not all of these adaptive loci certainly have been contributed. There's some really beautiful work by Feng Tian's lab. There's a large effect allele on leaf angle that was not found in modern germplasm, that when he included into modern germplasm could actually increase hybrid yields by almost 10% by really dramatically allowing him to increase planting density. So there are those alleles that exist, but also a large number of these alleles have already been incorporated into modern germplasm because maize breeders are good at their job.
Thanks, Jeff. Looks like we don't have any more questions in the chat. So with that, I'll give everyone maybe a couple seconds here if they have any more questions. Otherwise, I'd like to thank you for your time today. It was a fantastic webinar, and thank you for answering all our questions. I also want to advertise real quick. We did do a previous podcast with Dr. Ross-Ibara that is currently on our Breeding Bites NPP podcast that you can find on Spotify and YouTube as well if you want to learn a little bit more about Jeff. So with that, it looks like, yeah, I think we might be over a question. So thank you again, Jeff. And thank you everyone for joining our webinar. I hope you learned a lot, and I'm sure you could always reach out to Jeff with more questions.
Be happy to. Thanks all for coming.
Okay. Well, thank you, everyone. With that, we will conclude our webinar.
Thanks.
Thank you. Okay.